I'd like to, put your prediction hats on and think about, some of the other questions that you think we should be asking about the quality of our products and about AAV. We have, the full empty capsids. We've talked a little bit about partial. I think, Andrew, as well, you brought up a couple of other potential impurities or, questions that we should be looking at in terms of AAV in the future. But are there other purity or quality related questions you think that we as a field that should have the same amount of attention as full empty capsids do, right? I feel like full empty capsids get so much attention in this field as we're trying to learn. Are there others that you really wish had some of the same amount of star credibility that could steal some headlines? We can start with Justin here and go to you, Andrew.
I mean, I think for me is there's a lot of focus on the characterization of capsids, and as we noted there. But to me, one is the relationship of potency to the other characterization methods as we understand it. So, I think the better we can understand the influence of some of these deeper characterization technologies and measure them directly, and get to the potency, will help ultimately. And I think that, I said as we have more technologies, that story will continue to be written.
Yeah. Andrew, what do you think?
I would agree with the point that Justin made about potency, tying certain attributes to potency. For example, deamidation, for example, other capsid modifications. And then something else besides that I would be interested in is immunogenicity. So one concern is we know we've kind of established that CPGs in the gene of interest are undesirable, but an assay to measure immunogenicity of a particular capsid, how likely there's going to be an immune response. And then, so you have that, and then you can use that assay to link back to more basic quality attributes, deamidation, capsided DNA, VP1-2-3 ratio, these kind of things.
So really overall being able to understand how our quality attributes are impacting the function, right? The clinical function of our product, the immunogenicity of it, the performance of it. Yeah.
Safety, efficacy, immunogenicity.
Yeah. We actually had a question come in from the audience, from Rihanna here. With this recent slate of AAV approvals, do you see emerging focus from regulatory agencies outside of just empty full ratios? Do either of you have any thoughts on that? Have you heard any water cooler talk, anything through the grapevine about, what regulators are potentially turning their attention to as well, in addition?
Well, I would say comparability, which we already discussed.
I'll dig a little bit more into that here in our next set of questions. Justin, do you have a thought?
I think comparability, and then probably the state of the validated state of your methods, your understanding of your analytical methods that you're using. I think that's one that the industry and the agencies continue to push to have better and better understood analytical methods, specifically around content potency.
Yeah, and they're particularly focused on the strength assay. So VG titer, getting that qualified early, measuring the tox material with a consistent assay all the way through to the early stage to the late stage, and validating that before early stage trials. I think that's something that they've observed from perhaps some of the people that are now approved, that it was hard to compare their trials because they were using different strength assays in terms of VG titer.
Yeah, changes within comparability is challenging enough, but whenever content and potency methods change throughout the life cycle, it makes it a very big challenge to go through. And the technologies have matured, so I do see agencies pushing you for earlier and earlier understanding of those and characterization, so you don't have to have that overhang during your comparability.
Absolutely. Is it a matter too of changing your technologies, your assays throughout development? Is that part of the issue as well, in terms of as you're moving through development, comparability of the methods themselves? That's a challenge.
I think from 8 years ago, some of the initial products went on there using an animal model for a potency, right? There was a mouse potency method for one of the earlier products that went through development. I think that has probably moved back to the background more now, and you're more in vitro potency methods, and you're getting more towards, true function, better representative methods with the tissue of interest, et cetera, going into those. So I think, and in the years that has advanced greatly, and you're in a better state now with the program you're starting. But I'd say if you started a program 8 years ago, the state of technology made it much more challenging to manage their way through. And I give credit to the companies that make their way through the forest here to get those therapies approved. I mean, it's truly, I don't think they went through that. It's a matter of the technology they had available at the time. So you had to navigate your way through that. I hope that others of us can stand on the shoulders of this new technologies and make this easier on ourselves for programs going forward.
Yeah. We have another question here from Michael about aggregation, if that should be a concern for clinical dose. Do either of you have the answer to Michael's question in terms of impurity?
So in terms of aggregation, should it be a concern for clinical dose? I would say yes. You're required to have an aggregation measurement on your release specification for every batch. And then we could argue whether it's SE, HPLC, or DLS, or something else, or both. And is it a concern for clinical dose? Well, in kind of protein biotherapeutics, aggregation is a concern because it could lead to more immunogenicity. So you could argue that because AAVs are very immunogenic anyway, maybe it's not a concern, but then we've seen that we have these large kind of immune-based responses to people. So I think it's definitely something that should be controlled. And of course, if your product is aggregated at a certain point, that's going to affect potency as well, because it's probably not functional if it's all clumped together in a big mess.